Hi there, ladies and gentlemen. This is Mr. Workman, and this is going to be your screencast session 1 for the classical genetics unit. Just want to make you aware of a couple of key resources. Learning target list on page 7 of your unit packet, and 8 as well. Page 9 is a vocabulary list. The webpage DNAFTB, which stands for DNA from the beginning, DNAFTB.org is a good one. You're going to have class time to work through some of that information on that web page collaboratively with your classmates, as well as under the guidance of either myself or Mr. D or Mr. Gales. And then I think it's probably useful for you to consider doing some active reading and diagram analysis, pages 11 through 20 during class time. And then I also want to let you know that pages 41 through 50 in your unit booklet have some of the same diagrams and figures that you're going to see in this particular presentation. This particular screencast session presentation is going to focus on the work of Gregor Mendel, and just a little bit about the history of Gregor Mendel here. He is widely considered the father of genetics because he was one of the first to study the patterns of inheritance of physical characteristics. He was born in 1822 and he died in 1884. He was a monk in Austria for much of his life, and as the monk at the monastery who was in charge, or one of the monks in charge of the garden, he had a tremendous amount of time to spend in the garden, and he was noticing some things about the particular characteristics of peas, and he ended up being intrigued by that and studied how the physical traits of particular characteristics of pea plants would be transmitted from one generation to the next. And he ultimately put all of his observations down in a paper, and he wrote that paper in 1865. And so much of what I'm going to talk about today is based on his initial observations and conclusions as he studied the pattern of inheritance of particular types of traits. Now, you might ask, why peas? Well, one of the reasons why peas were convenient for Gregor Mendel is, first of all, they were growing in the garden at the monastery, but second, they're easily controlled in terms of their fertilization and pollination, and they have physical characteristics of varying types that are easily observed. So take, for example, this particular flower of this particular pea plant. Notice that the petals are purplish pink in color, okay? And I want to show you here, this is a white flower petal plant, and it's a pea plant, just like the other one was. It's just that you have a varying form of coloration of the flowers. Okay? So as you look at this, to understand what you did to study processes, you have to understand a little bit of the anatomy of the flower in a plant. Now, flowers are the reproductive structures of plants, and so they contain the structures that produce male gametes and female gametes. In plants, we more often than not call female gametes ovules, kind of analogous to eggs, and male gametes are called pollen, analogous to sperm. And a flower can self-fertilize because these structures, which are called anthers that produce the pollen, wind can push the pollen off of those anthers, or a pollinating insect or maybe a bird could rub up against those anthers, and pollen could be pushed from the anthers onto the stigma, which of course is the structure that receives the pollen. And if that pollen goes down through the stigma and were to fertilize these ovules, you get seeds. And those seeds obviously could be eaten for nutritional value, or they could be planted in the ground and produce a whole another set of pea plant. So what Mendel did in some of his experiments was control the pollination of plants, and he took it upon himself to control those fertilizations by removing, and you can see the scissors right here, he removed the male structures. I know, kind of cringe when you hear that. He removes the male structures of this purple flowering pea plant by literally cutting away the anthers before the flower is mature enough to produce pollen. And he took pollen from the anthers of a white flower petal plant, for example, and brushed that pollen from this plant onto the stigma of this purple flowering plant. And so therefore he produced what we call cross-fertilization. All right. Please recall that when self-fertilization happens, that's when a plant fertilizes itself. Cross-fertilization is when fertilization occurs between two separate plants. Now, going back to this, I need you to understand that self-fertilization is going to produce what we call true breeding generations. So if a white flowering plant fertilizes itself, the next plants will be white flowering plants. If a purple plant self-fertilizes, the next generation of plants that come from the seeds of that fertilization will be purple flowering plants. That's called true breeding generation. Mendel was trying to figure out what would happen if you did this cross-fertilization. And what he produced was what we call a monohybrid. Mono means one, and hybrid means a mixing, all right? So there's one trait that we're considering here, the flower petal color, and we're hybridizing the factors or whatever it is that controls those flower petal colors. Now, as Mendel did this, he got the peas that results from the pollination that he made happen, and he planted those peas, and the next generation of plants were all purple. And this was confusing to him initially, but he reasoned it out. Now, um, some other traits that he studied, of course, weren't just flower petal color, but the form of the seed, so the shape, whether it was round or whether it was wrinkled, all right? The color of those seeds, whether it was yellow or green. Here's our flower petal color. You can look at the color or the form of the pea pod itself, whether or not the pod looks full and plump or constricted, and whether or not the pod is yellowish or green in color. You can also look at the placement or the position of flowers along a plant, whether it's an axial position, so along the stem, or terminal position, or only flowers occurring only at the end of the stem. And sometimes you can look at the size characteristic of these plants. Sometimes they're really tall and sometimes they're not. So in addition to white and purple flower petal colors, Mendel looked at a host of particular characteristics that had two varying forms, and he did this cross-pollination in all those situations. Now again, what we're talking about here is Mendel cross-pollinating purple flowers with white flowers, okay? And in the next generation, you got all purple flowers. Now, here's a couple things to notice here. That P generation, that stands for the parent generation, okay? And then the next generation, so the babies, if you will, the children of those parents, this is called the F1, and that one is written in subscript. That means filial. So the first filial is the first set of pea plants that came from the initial cross. Now, this initially confused Mendel, as I referred to before, that all of the first filial generation were purple, because he knew that these plants must have something in them that would, control for the white color of flower petal colors. And what he reasoned was, because all of the next generation, or the F1 generation plants, were purple, he reasoned that whatever thing was controlling the characteristic, there must be a dominant form that causes the other form not to be seen. Now, when he let this generation, this F1 generation, fertilize, and he allowed it to self-fertilize, okay, so I'm going to do that little arrow in a loop, that means self-fertilization. In the next generation, which we're going to call the F2 generation, let's make sure we understand what this means. So if the P generation is generation 1, the F1 generation is actually generation 2, the F2 generation is generation 3. So think of it like grandparents, parents, grandchildren. All right? Hopefully that helps you. But anyway, in the second filial, or the F2 generation, the result was that he got some purple plants and some white plants. Now remember, this is purple plants self-fertilizing. And when Mendel saw that, he said, well, there's got to be something for the white flower petal color in this plant, again, because when it self-fertilized, some of its babies, the next generation, the F2 generation, had white flower petal colors. Now, if we do an analysis of these numbers, 705 purple plants, 224 white flowering plants, that's not immediately obvious what that means. But you got to understand here, Mendel did observations of lots of other traits. And what he started to conclude was that traits are passed on by factors. He did not know what genes were, okay? He called them factors. We know about DNA and how DNA is translated into protein structures, and that those protein structures ultimately cause our physical characteristics that we can outwardly see. But he didn't really understand that process, but he knew there must be something controlling the physical characteristics. He called those factors. And he also reasoned that factors have more than one form, all right? And he called those different forms alleles. That's a key term, all right? Now, and he also said that each trait must have two alleles, because how else could a purple plant, this is like our F2 cross, two purple plants, produce some white flowers. Now, this is what we call a Punnett square. Okay, and these Punnett squares can help us in predicting what types of outcomes we can have in a particular fertilization or pollination or cross that we could do. But let's go through what this all means, because you see all these letters here and you don't necessarily understand what they mean. So let's work with a little bit about what this all means. Let me get rid of these ink annotations here and just talk to you a little bit about how these alleles must be. Please recall that our DNA is found in chromosomes, and we have two sets of each type of chromosomes in a diploid organism. And what I want you to realize is that in homologous chromosomes, we have one set on each chromosome of all of our genes, but we have two of each type of chromosome. Please recall that. So what that means is, if we understand chromosome structure, and again, Mendel didn't know this, that's where our two forms, the two alleles, come from each trait. It's that you have an allele on one chromosome and you have another allele for the same physical, or for the same gene or type of gene on a homologous chromosome. And you could have two of the same alleles, or you could have different alleles. In this situation, we have an allele for purple flowers on one chromosome and an allele for white flowers on the homologous chromosome. Let's talk about how we might symbolize this to use those Punnett squares that we're going to talk about here. Please understand that when we use a symbol, we use letters, and we usually use the first letter of the word that describes the dominant trait. So, for example, purple is our dominant trait, right? White is what we would call the recessive trait. So we use capital P to represent purple, and we use lowercase p to represent white. We do not use W. Okay? We do not use W for white. So I want you to try a few. I want you to consider round seeds that are dominant over wrinkled seeds. What symbol should we use? I want you to consider yellow seeds that are dominant over green seeds. What symbol should we use? And tall plants dominant over short plants. What symbol should we use? Pause the video now. Think these through. See if you can figure it out. All right. Now, hopefully you're back and you figured out that you should use capital R for round, lowercase r for wrinkled. Again, not W. Capital Y for yellow, lowercase y for green, not G. Capital T for tall, lowercase t for short. This is how we use allele symbols when we're going to use our Punnett squares to figure out what can potentially happen. Another couple things to consider here. We need to define this term, genotype. All right? A genotype is, of course, the actual allele combination that an organism might have. It's the type of genes that are in the genome. Phenotype, on the other hand, that's our physical characteristics. Do you see how physical and phenotype start with the same two letters? That'll help you, hopefully. Now, what I also want you to understand is that we have three possible genotypes when we're considering alleles for one particular trait. You could have two capital P's together, you could have two lowercase p's together, or you could have a capital with a lowercase p. And we have particular terms that go with those particular combinations of alleles. Now, homozygous is what you have when you have two alleles that are identical. So that could be either homozygous dominant, as we call it, which is also referred to as purebred dominant, and then you could have homozygous recessive. See the two capital letters here? That's homozygous dominant, all right? Two lowercase letter p's here, that's homozygous recessive. The other possible combination that we have here is heterozygous. Now, just to talk to you about what these terms mean and what they come from, of course, heterozygous means different alleles, one capital, one lowercase. Let's break this word down. Homo means the same. And zygous, this word comes from the word zygote. And you should know what a zygote is. A zygote is the first cell that you get after fertilization. So in a homozygous situation, a zygote has the same types of alleles. Whereas hetero down here, this means different. Okay, there's different. And again, zygous is there. So we have different alleles in the heterozygous genotype. The other thing that you really need to realize here is how do the genotypes and how do the phenotypes correspond? Okay? Please realize that it is these two genotypes, the homozygous dominant and the heterozygous, which give us the dominant phenotype. Only when you have the homozygous recessive genotype do you have the recessive, or in this case, the white flower petal color phenotype or physical characteristic. This term hybrid again can be used to indicate that you have a mixing, as you see here, different types of alleles. All right, now let's consider this Punnett square. All right, this is like our P generation cross. We have a true breeding or homozygous dominant purple plant crossed with a true breeding or homozygous recessive white flower petal plant. And please recall that in meiosis, you have separation of homologous chromosomes and then separation of those sister chromatids in the second meiosis. And so what ends up happening is in a gamete, you can only pass on one form of a chromosome. You never pass on two forms of a chromosome, as is the case in mitosis, only one form of the chromosome. So in gametogenesis, which is the genesis of gametes, the cells go from diploid to haploid. When you look at two P's here, that indicates two forms of the gene. That's a diploid. This is a genotype. Genotypes are diploid here. When you go down here, this is the gamete possibilities that arise from that genotype. And these gametes, okay, from our purple plant here, these end up down here, okay? And in a Punnett square, what you do is you put the gamete possibilities on the outside of the square, and you just recombine them. You put the big P here with the lowercase p here, as is the case in all these different particular parts of this Punnett square. And please recall that when Mendel cross-pollinated white flowering plants with purple flowering plants that were true breeding, all of the next generation was purple. And this is why, because all of the next generation is heterozygous, and heterozygous genotype leads to the dominant phenotype. Let's do another particular situation here. Let me get rid of the animation. This has to do with Mendel's law of segregation and what we now associate as separation of chromosomes during meiosis. Let's say we have beetles here, and this is a fake situation, and understand here that this Punnett square process oversimplifies a lot of physical characteristics that we see in organisms, but nonetheless it is still useful. Let's say we got blue beetles and green beetles, and let's set up this situation. Let dominance be the blue color and let recessive be the green color. So if we have a blue beetle, that means that either the blue beetle could be heterozygous, as is shown here, or it could be homozygous dominant. A green beetle must be homozygous recessive, all right? And notice that we use the letter B here for the word blue, all right? We still have letter B's here, but lowercase b's. Now, if this is our guy, there's two types of sperms that he could make, all right? He could contribute sperm with the recessive allele, or he can contribute sperm to the fertilization with the dominant allele. There's a 50-50% chance of him contributing one form of the sperm or the other. So we put those different gamete possibilities down here on the outside of the square. Mama beetle here, she's homozygous recessive. The only thing that she can contribute to a fertilization would be the lowercase or the recessive alleles. And when you recombine those letters, in some instances you get the, that's getting a little messy here, let me erase that. In these instances you get the heterozygous genotype possible, and half of the others would be homozygous recessive. Okay? All right, now let's move on to this next situation. So here's a blank Punnett square, and what you're given here are gamete possibilities. And this just is a diagram that shows you how to fill them in. So for example, let's say we have two heterozygous organisms crossing, as is the case with the F1 generation self-fertilization. What you do is recombine these gametes in any possible fertilization. And as you can see down here, you've got 3 out of the 4 genotype possibilities leading to the dominant phenotype. This is homozygous dominant genotype, this is heterozygous genotype, this is heterozygous genotype. So those 3 would be purple, if we're talking about purple flowers, and only this one would be white. So we have 3 out of 4 possible purple, 1 out of 4 possible white. Now this up here is a different situation. So what I want you to do is try to put this down on a piece of paper and try to figure it out for yourself. We've got a heterozygous parent here. That's a capital P. This is a lowercase p here. And this is a homozygous dominant parent here. Fill in the square yourself. See if you can figure it out. Pause the video and do it. Okay, now I hope you're back. You should have filled it in like this. Capital P here, capital P here. Lowercase p, capital P, capital P, capital P, lowercase p. So half of our next generation in that instance would be homozygous dominant, half would be heterozygous, 100% would be the dominant dominant phenotype. So again, here's Mendel's work. He crossbred a parent generation, purple flowers, true breeding purple flowers with true breeding white flowers. The next generation, he got all purple flowers, and the reason, of course, is that 3 out of the 4 genotypes lead to, or 75% of the genotypes lead to the dominant phenotype um, in the next following cross here. Okay, I'm sorry, I think I might not have been clear there. In the first cross, all of these plants are heterozygous, so they are all showing the purple phenotype. But if you allow these plants to self-fertilize, you get what we had in the previous um, screen here, which is 3 out of the 4 possibilities showing the purple dominant phenotype. Okay. Now, let's look at this. This is a pea pod, just for example, a picture that was taken of a pea pod. We've got round, and we have wrinkled. You can see here that there are 9 round, and there are 3 wrinkled. And I did capital R for round and lowercase r for wrinkled. That 9 to 3 is the same thing as a 3 to 1 ratio. Okay? 9 is to 3 is 3 is to 1. And Mendel saw these particular types of ratios in multiple scenarios. So he did not just breed purple and white flower petal colored plants. He looked at a bunch of these other particular traits. And as you can see, he didn't always get exact ratios of 3 to 1. Sometimes he got really close. All right? This is pretty close. If you do the math, that's an almost 3 to 1 proportion right there. But if you add all this up and if you increase your sample size, the greater number of iterations that you do this type of thing, the greater possibility that your total results end up matching expected results. And this was the classic phenotypic ratio. When you do a monohybrid cross, a heterozygous cross, the next generation is generally going to be expected to exhibit a 3 to 1 proportion of dominant to recessive characteristics. Okay? Now, here's an interesting thing for you to do, pages 49 through 50 in your packet. This is a situation where you're going to look at tall plants and short plants. You're going to use the symbols capital T and lowercase t. This, of course, means tall. The lowercase t means short. What I want you to do is use those symbols and figure out how they go through a cross using Punnett squares on the visual organizer on pages 49 through 50. You should have that done for class, all right? Just as a practice work as you go through this particular screencast. So let's talk about Mendel's conclusions. We've already talked a little bit about the law of segregation, but I want to make sure that we point this out in order here. We alluded to this idea that one allele can dominate over the other, and I want to make sure that you're aware that we have this law of dominance that we talk about as it relates to Mendel's work. Okay? And this is a key idea here. The only way to see a recessive trait is if there are 2 copies of the recessive allele, which of course means homozygous recessive. Okay? That's the only way you see white flower plants here is if they have the homozygous recessive genotype, which of course would be little p, little p. Okay? These plants are either homozygous dominant or heterozygous. Okay? Here's the idea. Here's the second conclusion of segregation. The law of segregation, Mendel concluded that factors must separate somehow during the process of forming pollen and ovules, or what we call gametogenesis, the genesis of gametes. And we now know that as a result of meiosis. So that's anaphase of meiosis. Okay? That segregation happens as a consequence of the chromosomes, the homologous pairs, separating. So specifically, that's anaphase 1 of meiosis. Okay? Now, here's an interesting thing. Sometimes you see an organism that exhibits a dominant phenotype, but you don't know its genotype. If an organism expresses a recessive genotype, you know the genotype. It has to be homozygous recessive. But the problem can sometimes be is, do you have a true breeding, homozygous dominant organism, or do you have a hybrid? And you can do a test cross to test this out. And what you can do is test this unknown genotype organism with a known genotype, namely the recessive phenotype displaying organism, and you look at your results. If you get all plants in the next generation that are purple, you know this plant must have been homozygous dominant. Okay? If you get half your plants white, or thereabouts half your plants purple, then you know your plant must have been heterozygous. Okay? Now, that's it for this particular screencast. Mr. Gales is going to talk about, or maybe I will talk about, not monohybrid crosses, but dihybrid crosses in the next session.